Guys, we're gonna start with our quiz today, and then we'll have a little bit of time for a final review before our exam on Monday. Don't forget that the exam is scheduled for 1 o'clock in this room on Monday, from 1 to 3. All right, there we go.
That's not helpful.
question. Which stage of the Plasmodium protozoan infects red blood cells of humans? All right, so we have schizoid, merozoite, sporozoite, and chlamydocyte. So, merozoite, right? That's the one that's in the red blood cell. So, what is the form that infects or is transmitted by the mosquito? Right, sporozoite. So it's really important to keep those two straight and remember those terms. So, most promising method to control malaria is bed nets, right? We talked about that. So there's been some investment in vaccines. The vaccines aren't highly effective yet. We have some drugs, but again, there's resistance that has arisen to the drugs. So most countries are going to invest in bed nets because they're the cheapest and most effective. Why are bed nets super effective? Right, because that Anopheles mosquito only bites at night. All right, CRISPR-Cas9 system used for genetic engineering. Cas9 is an endonuclease, right? That's how it works. Makes double-stranded breaks in the DNA. You can modify it, right, to make one break, so to become a nickase. How else can you modify the Cas9? What could you use it for? So I'll give you a hint. These double-stranded breaks, if you use a single Cas9 enzyme, right, the cuts are right across from each other, right? So you have blunt ends. What other kinds of cuts could you make with Cas9? Right, staggered cuts. So you could use two guide RNAs to make staggered cuts. What else? There's one more thing we talked about that you can do with the Cas9. What could you do with it if you had a guide RNA but you had mutated both domains? Well, if you mutate them both, it's going to be a dead enzyme, right? But what else could you do with that dead enzyme? Yeah, you could basically use it as a guide to tag region. So you could put another effector domain on it, something like a GFP tag, or another enzyme, one that we talked about is a methylase, right, to methylate the DNA. All right, tetanus-spasmin intoxication causes what kind of paralysis? Rigid, right? Flaccid paralysis is caused by botulinum toxin. All right, and the last question. It's usually beneficial to add nitrogen to stimulate hydrocarbon degradation. We talk about hydrocarbons or hydrogen and carbon. So if you add a source of nitrogen and what else? Phosphorus, right? So you want to fertilize those bacteria to keep the degradation process going. All right, so I wanted to review some of the basic information that we're going to cover on this exam. So we're not going to talk about the viral diseases or the protozoans or the fungi because we've done that already. So what we're going to do now is just going to talk about all the other stuff. So there are some questions that I told you guys when we started this unit on the exam that are general about viruses. So remembering that they're intracellular parasites, that they can have DNA or RNA. And what's important about the DNA or the RNA? What does the DNA or RNA virus affect? Yeah, exactly. exactly. So the replication or multiplication of the virus, so the stages that happen. And if you're an RNA virus, what important enzymes are you going to bring along? RNA polymerase, right? And if you're a DNA virus, you're going to bring along DNA polymerase. And remember, that's really important because when we talked about acyclovir, for example, remember, acyclovir is a nucleotide analog that can't be extended from, right? It looks very similar, and it gets phosphorylated by the viral-specific kinases, and then is used by viral-specific DNA polymerases. It's not used by the polymerases in the host cell. So bringing along viral-specific DNA polymerases is a really important aspect in terms of targeting mechanisms of the treatment. So we talked about viral size, we've talked about morphology, we've talked about envelopes and spikes. So what do you think you need to know about viral spikes? Yeah, they're for attachment. And what specific example did we talk about? HA and NA. And what's important to know about HA and NA spikes? Right. HA is used for attachment. NA is used for release. And what did we learn about related to that? Related to the NA, the neuraminidase spikes. A drug, right? Which drug? Tamiflu. That's right. So we learned about Tamiflu. So there's one more concept. Why do you have to get the vaccine every year? Right. Right. The changes that you see, the antigenic shift and the antigenic drift. Right? So remember, drift are small changes, shift are large changes. Genome rearrangements due to two or more viruses being in the same host cell. So that kind of aberrant packaging. All right, so again, you should review the basic information. You should review life cycles, really focusing on what we just talked about. DNA viruses, their DNA goes to the nucleus. We have DNA polymerases that are brought along. And then RNA viruses, the RNA stays where? In the cytoplasm. And how do we differentiate? What's special about plus strand viruses versus minus strand viruses? Right, plus strands look just like the mRNA. They are sense RNA, so it makes sense. It can be translated right away. All right, then we also talked about another class of viruses, the retroviruses, and retroviruses require what enzyme? Reverse transcriptase. Now what's the example of a retrovirus that we talked a lot about? HIV. So what do you think are some important things to know about HIV? Yeah, so you should know the disease progression, and we've talked about a lot of secondary infections, or then what happens to HIV/AIDS patients because they're immunocompromised. A lot of the opportunistic infections that they can acquire. But in terms of the viral life cycle, what's really important to know, besides it's a retrovirus? What else? Yes, so attachment, right? That's really important to know. So, CD4 positive cells, those are helper T cells, and what in addition to CD4? CCR5. So you should remember those two things. The virus attaches to cells, so it can also affect like macrophages, but it's the helper T cell count that when it drops, makes individuals really susceptible to other diseases. And of course, we've talked about the CRISPR babies and the attempt to gene edit that CCR5 locus, right? To give HIV immunity, to make them immune to HIV infection. All right, so some other terms that would be really important to recognize. We've talked a lot over the semester about latent infections versus acute or persistent chronic infections, right? And remember, acute infections show up right away, right? That's what acute means. We have lots of examples, especially with the viruses, of infections that can show up weeks, months, days, years later, right? What's an example of latency, a viral example, a latent virus that we talked about? Yeah, shingles, right? HIV, hepatitis. Lots of examples of latency. So, again, we're not going to talk a lot about the organisms today, so hopefully you're reviewing them, and don't forget about the gram-positive, because like I mentioned a few times now, you're going to see a similar question to what you saw on the last exam about the gram-negative in short answer form, virulence factor, how it contributes to disease, and you'll have a list of examples. So don't forget about that, and you'll see some multiple choice questions about the gram-positive. So what I wanted to talk about for the rest of class today was really review those things that you're going to see some more short answer questions about. The cocoa fermentation that we talked about last class, you'll see a multiple choice question about that, so that's not on the short answer list, it's a multiple choice question. So kind of file that away. But in terms of the short answer questions, we're going to talk about the CRISPR-Cas9 today. So that will be a multi-part short answer question, kind of guiding you along. Things that we talked about when we were reviewing the quiz, those are important things to know. You need to understand how the CRISPR-Cas9 system works, what it is naturally for in the bacteria, how it can be modified for genetic engineering purposes. You will also see a question about the tetanus versus botulism. So tetanus spasmin versus botulinum toxin. Those two toxins, remember, you're going to be able, hopefully, to compare, contrast. We'll get to some of that information today. There's also a question about Bt toxin. So we'll review that specifically how Bt toxin works, affects insects, affects the gut of the insect. And then biofuel, so comparing, contrasting hydrogen and ethanol. Hopefully you can list 2 advantages and 2 disadvantages for each. And then bioremediation, that one slide that we talked about last lecture, things to consider, like fertilization, dispersants, predation, things that can help and also things that can hinder degradation when we're trying to use an organism to help degradation for the bioremediation. So there's 6 main topics: gram-positive, the Bt toxin, the CRISPR-Cas9, biofuels, tetanus versus botulism, and bioremediation. Those are the 6 topics that you'll see in the short answer section. You'll see a handful, about 5 fill-in-the-blank questions. So there have been a lot of terms that we've reviewed, things that you should be able to recall, information you should be able to say given, a sentence and some hints. And then the rest will be multiple choice. So let's start with the CRISPR-Cas9, CRISPR-Cas9 system. Anybody have any questions about CRISPR-Cas9? Yes. I am going to go over it. But a lot of you, when I said, how many of you guys have learned about CRISPR-Cas9 before? Like, over half of you raised your hand, okay? So, any questions? Everyone understands it? No. Okay. That's why I'm asking. Okay. So, first of all, if we think about the components that are needed. Now, you guys told me why already we have the CRISPR-Cas9 in lots of bacterial species, like streptococcal species. Why do they have it? It protects against viral infection, right? And it's an immune system. And what's cool about it is it's not just a general immune system. Like, that's why they have restriction enzymes, right? That now we use for genetic engineering. They have restriction enzymes that just cut up randomly. What's special about CRISPR? Yeah, it's a memory, right? So they see a virus, and part of that gets coded in a memory here in the CRISPR array. So between these protospacers, the little black boxes, you have short intervening sequences here that are invader DNA. And so the CRISPR repeat space array gets transcribed, so you get these RNAs that will then be processed and mature into a guide RNA with the help, of course, of the tracer RNA or the transactivating RNA. So you have to have an RNA transcript from here and your tracer RNA and RNA transcript from here. They come together. And then what do they guide? The enzyme, right? And so there are several endonucleases in this Cas operon, but Cas9 is the one that is utilized for genetic engineering purposes. So that Cas9, what's special about the Cas9 enzyme? So cleaves DNA, so it's a DNA endonuclease. And what two domains does it have? These two nuclease domains, right? The HNH domain, and that part of the Cas9 is going to cleave where? It cleaves in the complementary DNA sequence, right? That's complementary to the guide RNA. And then the RUVC-like domain cleaves where? The other one, that's right. Opposite of that, right? So that's what's key. You have these two endonuclease domains that recognize specific consensus sequences, okay? And are going to recognize, be brought to the DNA and bind to it, guided by this guide RNA, right? That's what leads it to a spot. And then the HNH domain cleaves on the complementary strand, and the RUVC-like domain cleaves the other strand. All right, so that's the genetic components and then the transcription product. So if we look at the way that the Cas9 enzyme works, again, you have to have the CRISPR RNA, you have to have a mature tracer RNA, and they bind to the Cas9 enzyme, recruit it to a region of DNA that's complementary to that tracer RNA sequence. And then you also need what? What's key? So you can't just have a sequence that's complementary to DNA. What also is necessary to get binding at that region? The PAM. So in your answers, you need to talk about the PAM, the protospacer adjacent motif. So remember, it's, pretty common to find any nucleotide GG, right? And for eukaryotes, there's a C substitute. But for any organism whose DNA you're going to cut, there is this protospacer adjacent motif that must be there in order for effective cutting. So we have our PAM, we have our guide RNA, we've got a complementary sequence. Now our H domain is going to cut that complementary strand. The RUBC-like domain is going to cut the opposite strand. And you get a double-stranded break. So what's been done to use it for genetic engineering purposes? Okay, so we've modified the RNAs, right? So instead of supplying DNA to make a CRISPR RNA, or the tracer RNA, and supplying DNA to make a CRISPR RNA, you supply a DNA sequence that's modified with a few nucleotides here to make a linker loop region. Right? It just makes it more efficient. So we have this linker, and that gives us a single guide RNA. So instead of having a CRISPR tracer RNA duplex for genetic engineering, we use the single guide RNA to guide the Cas9. And then we talked about once you cut the DNA, what your desired outcome might be. Okay? So you make a double-stranded break, and cells can repair that through non-homologous end joining. So let's say that you had a gene that was repeated in the genome through gene duplication, and it was making too much product. And so you wanted to get rid of just one copy. You might just use the non-homologous end joining. Because through non-homologous end joining, once you make that double-stranded break, you're going to have a little bit of chewing away, and so it can create a gene disruption that way. Now, if you want to do more complex revisions, like insertions or truly edit, like make a single nucleotide base change, that you're going to have to supply a short sequence of DNA for homology-directed repair. Okay? So homology-directed repair is more complex because you have to add an additional segment of DNA that has the sequence that you want to insert or correct, and it's more rare, right? So non-homologous end joining happens throughout the cell cycle. Homology-directed repair does not, because it only happens when you have the homologs present, right? So at the start there, before cell division. All right, so we go back. We've talked about the loop. And the last part of the question is, of course, what you can use besides just a wild-type enzyme to make double-stranded breaks. What can you do with the Cas9 system? So you can create a nickase. This can encourage, again, homologous repair. So you do that by mutating one of the domains to make the enzyme dead on that side. You can use two guide RNAs to make a staggered cut, right? Instead of just that blunt cut, double-stranded break, you can make a staggered cut. And then you can make both enzymatic domains dead. So mutate the HN, the RUBC-like domain, and attach an effector domain. And we talked about a couple of examples already that they discuss in this review article. So adding methylation effector domain or creating a GFP fusion so that you can visualize regions along a chromosome that have the sequence that you're interested in. Okay, so what questions do you guys have about CRISPR-Cas9? Everybody gets it. Yes?
So downstream of the Cas9, it's photospacer and the viral DNA in like succession.
Yes.
So what's the difference between what's the difference between the CRISPR RNAs and the photospacer? Is the photospacer just
The protospacer doesn't give an important transcription product. Basically, it's a repeat sequence. So the reason why it's important is because it's part of how it was discovered. When they're sequencing the genome of these organisms, they were like, well, look, there's these weird repeats, right? The protospacers are repeats. They're like junk DNA or satellite DNA that we have all throughout our genome. But then when they looked in between these repeats, what they saw were sequences that were what? Are they bacterial sequences? No, they're bacteriophage sequences. So basically, they're just there so that as you get, and this isn't talked about in depth, how the CRISPR repeat spacer array continues to grow, but as you get viral infection, right, new sequences get added. So the repeats are important for that. If you think about like insertion sequences for transposons, they have those flanking sequences that allow for insertion in their repeats. Kind of similar in concept to that. Any other questions? Yes.
Go over the different tests one more time.
You mean the modifications or just the two domains?
So like the wild type double stranded break,
Yeah, so things that you could modify the Cas9 to do, right, besides just making a double-stranded break. You could mutate one or the other domain. So if we mutate the RUBC-like domain, eliminate that, only the HNH domain is active. If we mutate the HNH domain, only the RUBC-like domain is active. And there can be a variety of reasons why you might want to make a nickase, but one reason would be because by nicking here, that single break is going to be detected, and then cells will try to repair it. Sometimes with, again, homology-directed repair, so you could use a nickase to introduce a new mutation there, or to fix a mutation, you could try. Staggered breaks, by using two different guide RNAs, and then mutating both domains so you don't get any cutting of DNA, but you use Cas9 binding to label DNA in some way. All right, any other questions? Okay, so biofuels. What are the two biofuels you need to know? Ethanol and hydrogen. What are some advantages of ethanol? You can mix it with gas, right? So you can store it, transport it, mix it with gas. What about disadvantages? There's a few different disadvantages. Can make a lot of waste. It absorbs water, so you have to distill it twice, right? If you transport it or pump it, you have to, so if it's pumped through lines, it can absorb water when it's being transported through lines. So you have to eliminate the water twice, and it's not carbon neutral, right? So the desire is to get away from corn, right? The food portion of corn, because also another disadvantage of ethanol is that by using corn as a source, you can drive up food prices. All right, what about hydrogen? What are some disadvantages? It will go kaboom, right? Highly combustible, so you need to have, it's challenging, transport, storage, challenging, but what are some advantages? Right, it's carbon neutral, very energy efficient, right? You get the biggest bang for your buck with hydrogen. So if you can list some disadvantages, advantages, you'll be in good shape. All right, then we talked about Bacillus thuringiensis, Bt toxin. So for Bt toxin, you should know the organism that it comes from, Bacillus thuringiensis. What is Bt toxin used for? It's a pesticide, right? So it affects what? Insects. So, key things to remember. There are lots of variations in the Bt toxin, in the crystal proteins, the cried proteins. And they are very species specific for different species of insects. Why is it insect specific? Why doesn't it have an effect in our gut? Yeah, there are two reasons. Receptors in the insect's gut and the pH of the insect gut. So those two things. Insect-specific receptors that it binds to and the insect-specific pH in the gut. So when it binds to the insect gut, remember what happens is that these proteins essentially form a pore which allows for water and cations to flow in and ATP to flow out. So ATP flowing out results in what? What does it do to the insect gut? Paralysis of the insect gut. So they starve to death. Now, what's going to happen if a bunch of water is flowing into those cells? Explosion, right? So you get cell lysis. All right, so bioremediation. I'm gonna flip ahead to this picture. So we've talked about this a few times. Everybody feel comfortable with things that can increase degradation at an oil spill using agents for bioremediation, using bacteria, and things that could be harmful, right? As long as you can list a couple of things that could speed up degradation and list a couple of things that could decrease the rate of degradation, you should be in good shape. So review this figure, this portion from your book. All right, so cocoa fermentation, since we just went over this, I just want to point you in the right direction. Again, it'll be a multiple choice question. It will not be a short answer question. But we talked about the stages of fermentation and what's beneficial for producing good cocoa flavor, enhancing the flavor, and what is harmful. So if I were to label these A, B, C, D, E, which peak is most important for enhancing flavor? C, that's right. So the acetic acid bacteria are very important because then that's what's going to help, their products are going to help activate the hydrolytic enzymes within the pod. But you need the yeast to make products to then encourage the lactic acid bacilli, and you need the lactic acid bacilli to make products to encourage the acetic acid. So it has to happen in a specific order. Now, which peaks are most harmful or can result in off flavor? D and E, right? So the spore-forming bacteria, which genera specifically that we've talked about? The bacillus, right? So just like bacillus can result in that ropiness and make bread go off, bacillus can affect cocoa fermentation, and then the filamentous fungi also can produce products. But the bacillus produce a wide variety of volatile products that can make an off flavor. All right, so that is about it. The one thing that we haven't talked about is Botox versus tetanus. guys know, flaccid paralysis, rigid paralysis. Okay, so which organism causes tetanus? Clostridium tetani. Which organism causes botulism? Clostridium botulinum. Now, portal of entry for tetanus. Wound, right? Through the parenteral route or wound. What about for botulism? Portal of entry. Ingestion or a wound, right? But typically, ingestion of the spores. All right, so tetanus is rigid paralysis. Botulism is flaccid paralysis. Both of these are what types of toxins? They are exotoxins. They are also what type of toxins? Neurotoxins. And what else? AV toxins. Okay? So both of them are very similar. They're actually quite small, so it's kind of nifty how they work. So, cells that are affected. For tetanus, what cells are affected? Inhibitory neurons. Inhibitory neurons are affected. The inhibitory neurons produce what? The inhibitory neurotransmitters. And what are the two inhibitory neurotransmitters? GABA and glycine. How are those two inhibitory neurons affected? Yeah, so inhibitory neurons are in what that gets released at the synaptic junction? A vesicle, right? And remember, we talked about what proteins that are important for vesicle docking and fusion. The snares. So, what is cleaved, right? These are zinc proteases. Both tetanus spasmin and botulinum toxin are zinc metalloproteases, so they cleave protein. So which protein, which component of the snare complex is cleaved by tetanus spasmin? Synaptobrevin. So if we go forward, you should know these figures, right? Synaptobrevin. These are the key components you need to have in your answer. So for tetanus, synaptobrevin is cleaved, preventing or blocking the release of the inhibitory neurons, GABA or glycine neurotransmitters, so that the motor neuron that's stimulating muscle contraction just keeps releasing acetylcholine. Now, if we talk about botulism, botulism affects what? First of all, which neuron is affected? The motor neuron is affected. What neurotransmitter is affected? Acetylcholine is affected. Acetylcholine released because what component of the snare complex is cleaved? SNAP25 and syntaxin. So that complex, the SNAP25 syntaxin complex is cleaved by botulinum toxin, so acetylcholine can't be released. All right, any questions? All right, guys, well, good luck in your preparation. I will see you on Monday at 1 o'clock. The exam is your typical 50-minute exam. So it should be very doable. We have your 6 topics, a handful, about 5, fill in the blank, and multiple choice will end up somewhere between 30 and 35. So just like all the other 2 exams, all right? I will see you guys